Thanks, Susan and Andy and the organizer for inviting me to present my work here in this conference. I rarely talk about this subject ever. So today I'm going to really talk about phosphorylation mechanism by protein kinase to an unusual family of substrates. So these are SR proteins. So SR protein, as the name suggests, the serine arginine rich protein. So SR protein are the splicing factors. It's almost like transcription enhancers. These are the splicing enhancers. So there are roughly around 10 SR proteins in humans. In each there is no SR protein. So they are SR-like proteins. So what is the characteristics? That all SR protein must contain at least one or two RNA binding motifs. This is the RRN that they are known as. And then the C-terminus contains a segment, the region serines and arginines. And if you really look at it, they're really in alternate positions, serine, arginine, and so on. So what these SR proteins really do, that they're very important for this spliceosome formation. And so how do they do it? They actually bind to the pre-mRNA sequence called exonic enhancer element, EAC. So that in this is the sequence, a 7-nucleotide long RNA sequence is present within the exons. And by binding to the exons, they really assemble these HNRFPs and form the spliceosome. And phosphorylation, it's known that phosphorylation of the RH domain actually is very important. It affects the spliceosome assembly in both constitutive and alternative splicing. It's important for the intranuclear and internuclear localization. It's also important for mRNA export. So after splicing, they also play a role to export mRNA out of the nucleus to the cytoplasm for translation. And so this is the family of SR protein kinases, SR protein 1 in particular, which is sort of well characterized. And what is its characteristics? Its primary characteristics, of course, it has a really long segment that is sort of bifurcating these two kinase domes. Okay? And it is, the spatial domain is 250 days to be really long. This is really long. It's really its function is known as regular distribution of the SR protein and pre-mRNA splicing by phosphorylating, as I said about this SR protein. And it's really role in anti-cancer drug sensitivity, virus replication, and apoptosis. And then it's really known right now that SR protein kinase 1 and 2, they're really two isoforms. They are abnormally expressed in almost all cancer. What it really means, we still don't know. So obviously they really affect splicing, but what is the which pre-mRNA that really they affect in different cancer is not really clear. So what we really know so far about the SR proteins is spliceosome assembly, and this is really a black box. We really don't know. Why we don't know? Because most of the people don't work on it. So many people work on kinase only, or microRNA. You know, the splicing field is completely sort of lost all these different emergence of microRNA. So what do you really know that? So very like exon, this is the intron, the splicing reaction is the removal of intron, right? The sequence between exons. So what SR protein does, actually, it really is involved in every step, but we really don't know exactly how and what they do, how they really assemble it. But if you really think about spliceosome formation, this is the most amazing enzyme that ever existed. Why? Because this is the only enzyme to form on the substrate, and then it carries out the single round of catalysis and then disassemble. Right? And the formation is multi-steps, very, very dynamic. Right? So all of those things, the rearrangement of the RNA, coming and going of proteins, and really the one critical factor is SR protein, and the phosphorylation play a very important role. Both phosphorylation and dephosphorylation during splicing is very important. So it's not like it's just only one stage is really involved. As the spliceosome formation is happening, this SR protein undergo cycles of phosphorylation and dephosphorylation. And really this is the main thing that hopefully try to understand and we'll understand someday. So just to really just put it in the context that SRPK1, that if you really compare it to the PKA, that, as Susan mentioned, that PK has these regulatory segments, the N-terminus and the C-terminus. ASAPK1 has it in the middle. So we did the structure of a version of ASAPK1, which is active, constitutively active. What you have to do is actually remove most of this insert part. You have to really leave a small portion of insert, so in order to get crystal. And we understand it why we have to really keep it. So this is the segment, this green and yellow segment that is coming from this picture. So out of this 250 amino acid, we had to include roughly 40 or so. We still had to remove. Huh?
How did you know which 40?
To experiment. Graduates went really do all sorts of deletion, construction, all those things. So that's the way. So it took time, years, and this part is sort of the unique feature. This is we call it MAP kinase insert, which is present in MAP kinase family. It's a small lobe, and you can really see this activation loop with this active conformation and everything. So this is really poised to carry out catalysis. So we're trying to understand the mechanism of catalysis. That was our goal in collaboration with Joe Adams. So what we really did, tried to show a peptide, a substrate peptide. Our hope was the peptide would bind to the active site, and it did. What it really bound actually is bound to this close to the area. The bottom of this kinase is near to MAP kinase insert. So it was really puzzling, and obviously we didn't know what was going on, so we could really see it in here, and it was making all, the really right hydrogen bonds, the right complementarity of the interface. So we really knew that something has to be real, and but it is a substrate peptide. It should really actually bind to the active site. So we went on and showed actually that that within ASF there's a segment right between this RRM and RH domain that could actually mimic that the peptide, the substrate peptide we used for shocking experiment, co-crystallization experiment. And we did more experiment to show actually this is true. So this is the part of ASF where you would bind into the bottom of the kinase. So we really are excited so that we figure out a mechanism. So this is a docking site, the docking motif of the substrate is binding to the bottom of this kinase, and that really put this first serine in the proper position so that all this serine could be phosphorylated, right? So what I really didn't tell you that of this serine in this SR domain, that we can really now divide into two parts, SR1 and SR2. SR51 phosphorylates the first 12 or so, and in a processive mechanism, and Joe will talk about it in his presentation. And so we thought like this is the part that will bind to this kinase docking group, and then that will allow the phosphorylation sequentially. So this is another kinase that binds to the substrate with a very high affinity. It's at 50 nanomolar. And so we tried to co-crystallize the substrate. So again, we had to really make all these fragments in order to figure out a construct within this ASF which contains the second RRM domain and this RH domain. All right. So this is the fragment we use for the co-crystallization. This is actually NS3. Basically, it has most of these space are removed, and we got the structure. And this is what the structure looks like. So you can really look at the RRM2 as binding, sort of making contact with both the small lobe and the large lobe. Basically, the kinase structure doesn't undergo any changes. Very, very minor changes. It's very similar to what we really see in this unpeptide bound. So we have different structures of the kinase. There's no difference. And then we really see that the part of the peptide is really binding in here. So what is the most surprising thing was actually, and I'm going to really show you in the next slide. So there's the third feature we actually see. There's a phosphorylation is binding to the P plus 2 pocket. And because of the lack of connectivity, we really didn't know where it is coming from. Okay? And so this dotted line actually shows a lack of connectivity. So, but you have really very good density in here. This is not a very high resolution structure, 2.9. But we knew that which portion of this protein is actually is binding in here. And that was really quite surprising. Why it was surprising? Because actually this is not the part. This part is a beta 4. It's actually part of the secondary structure, which we thought originally that would be the docking motif, right? So that's beta 4 in here. And what is binding in here is the first part, the first 8 or so residue within the RH domain, meaning the part that will eventually be phosphorylated is bound in here. Okay, so that was puzzle, and I still remember this. When my student, two of my students actually came to my office, this like everything we said in the old page is all wrong, and that doesn't really make sense, and all sorts of things. They're very nervous, and I just laughed. I don't know why. I should be nervous too, but it's fine. So anyway, that's what we're really showing is beta 4 and all those things. So and if you really compare the old structure, the peptide bound structure versus this structure, they're very similar actually, exactly. So what is the important feature is that it's like arginine in alternate position. In both cases, the peptide substrate we use, or in this case of this substrate, the ASF complex, that so the arginines are really making contact. So this is really a charge-based contact. And if you really look at it, I'm going to talk about it. So first of all, we really had to do this experiment to see what's going on. Is that really what we proposed in our original structure, the peptide bound structure, the mechanism of phosphorylation, the docking motif? Is it really what's happening? Because we actually had a model. So maybe I'll just show you the model first, okay? And then we'll really explain this and go back and explain it. So this is the model. The model is, so here you have the kinase, okay? And this is the substrate. And this is the part that will be phosphorylated, the entire RH1 motif. And we really colored it into two different colors, the blue and the green. So the blue part is the one actually is binding to the docking group in the kinase. Right? We don't see any density of the green part. So even though we have the entire RH domain in there, in RH1 in the crystal, but you don't see the density. So what you really see is, what you really think is that the first shearing, so the directionality of phosphorylation has to be from the C to N terminus. So by part of the substrate binding to the docking motif, we put the C terminus of the RH1 in the active side, and it undergoes phosphorylation. The shearing gets phosphorylated and quickly binds to this pocket. Right? And that's very important to really have subsequent shearing to be phosphorylated in a processive manner, right? So the processive phosphorylation means all shearing must be phosphorylated before the product gets dissociated. It's not distributing. It's not like every shearing is phosphorylating and dissociating. And that's how this dissociation. So as it's moving, so this is a mobile. It's really as it's moving, beta 4 has to really melt. And when the first shearing undergoes phosphorylation, then the beta 4 actually is melted away, and now it's binding to the docking motif. So basically, the initial docking motif, and then the basic entire RH domain, and the beta 4 is really the docking motif, and it's just fitting to the docking group. And we actually did it. We proved it by chemical cysteine cross-linking experiment. And it's a little bit complicated slide. All what I would really say that in the presence of ATP, if you have the 10 micromolar ATP, so you really go ATP so that the entire RH domain will be phosphorylated, so that you really still get stuck. So what we're really showing here, so here is a lysine from beta 4, 193, right? So that will be part of the beta 4. So before any phosphorylation, that should not undergo any sort of cross-linking between kinase and substrate, right? And only when you have phosphorylation is happening, so that it will eventually come and melt from its secondary folded structure, and then it will really cross-link. And this is what we really see. So that you can see the efficiency of the cross-linking in the presence of ATP much more than in the absence of ATP. If we compare that thing, that 204 is part of this the RS domain. So that would really bind there right away. So this is really binding even before addition and NP. So we have to really compare that efficiency of cross-linking, and this is sort of the control. So that's what sort of led us to believe, and we did more experiments. Joe's lab did more experiments that beta 4 actually undergo melting in the presence of ATP. Okay, and we did even more experiments, and that's what's really happening. And so, and this is what really showing is that sort of the priming site. So the first serine must get phosphorylated, perhaps in a little bit slower rate, and that we allow this docking to the prime site, this pocket, and so the subsequent serine phosphorylation will really happen in a much faster way. And this is what really showing in this sort of charge distribution. If you really look at it, the SRPK1 is all really negatively charged, the kinase. And this is the part that, you can see the density of the peptide bound portion. But you can really see it is going all the way. Except there's a little small patch of blue. And that's the part where the phosphorylation is binding. Right? So, and so that's the characteristics of the kinase. So, there's something very unusual in here. So what we really knew from Joe's experiment was that RS domain, if you really just have the RS peptide, it's actually phosphorylation is distributed. Okay, it can still phosphorylate it. It doesn't have any restriction to phosphorylate all serine, but in a distributed manner. So that actually says that RRM, binding to RRM is doing something. So that is responsible for processive. Something in the process of phosphorylation. If you really look at the interface of this RRM to the kinase, it varies roughly 12 to 1300 square angstrom. But it's only 4 residues that are involved, and 4 or 5 hydrogen bonds are really formed between this RRM to the kinase. So we really tried to investigate a little bit more that what really RRM is doing. And we did the pull-down experiment. Again, I just will simplify it. So the basic result is that if you really have just this RS peptide, GST tagged RS peptide, it binds to the kinase. It can really pull down the kinase in very high efficiency. So RS peptide. So RS peptide actually binds very, very strongly, so nanomolar affinity. But if you really do the RRM, it really doesn't. Basically, just the RRM alone has absolutely, I wouldn't say no affinity, but it's very, very weak affinity. Right? So that was really surprising because if you go by, the way we really think about it, that if you go by this buried surface area, that the RS peptide portion is really bearing only 900 square angstrom versus this is 1200 square angstrom. It really doesn't make sense. But again, I didn't really tell you this particular this line. So what I'm showing in here actually, RS peptide binds to this kinase domain very strongly. RRM doesn't bind to the kinase at all. But now if you add peptide and then RRM and try to pull down to the RRM, they bind very strong. So basically, my slide is messed up. But this is the story. The story is that in the presence of RS peptide, the RRM binds very strongly. It's actually almost like stoichiometric affinity. Alright? So this is really coupled interaction. That something is happening when the RS peptide really binds that really allow this fixation of the kinase and so that really binds stably. And so what we haven't really done this experiment and we're really going to do it in collaboration with Joe, that if you really have this RRM already present in the reaction mixture, then the RS peptide would really probably be phosphorylated in a processive manner. So just RS peptide, even though it's bind very high affinity, but its phosphorylation is distributed. But in the presence of the RNA, and this is not what I'm really showing the processive phosphorylation. All what I'm really showing in here, this is this experiment that we just recently done. The student has done it, and this is just this RS peptide and the SRPK1 phosphorylate, and this is in the presence of the RNA in France. That you can really see. This is not a processivity asset. This is just a phosphorylation efficiency. The slightly enhanced phosphorylation efficiency you see. And we're going to really do the processivity experiment. So we believe that RRM really, it's just, RS domain, it really regulates its own phosphorylation in a very unique way. So obviously it has to be phosphorylated in a processive manner. It binds to the docking group, and it really recruits RRM to the bind so that phosphorylation can happen in a processive manner. And why all of this thing is important in cell is we still don't know. The why it has to be processing phosphorylation. But I think it's the rapidity, because if you really break open the cell, you never see any SR protein in the cytoplasm actually. In a sort of steady state, you see all the SR protein in the nucleus. So that means the proteins get synthesized in the cytoplasm, rapidly get phosphorylated, and all what it really requires are 10 or so serine to be phosphorylated in order to kick it into the nucleus. And then something happened in the nucleus. So basically the SR protein actually sort of stuck in the nucleus in a nuclear body called speckle, nuclear speckle. Right? And this is sort of this punctate in here, that's the nuclear speckle. And if you really overexpress this another kinase, which is clip-style, which is a nuclear kinase, then you see a distribution throughout this nucleus, so really this SR protein breaks out of this patient. So we did a lot of experiment, obviously this cell-based experiment, and then tested again the phosphorylation state of this protein, and then we concluded that this RS2 is the part that undergoes phosphorylation by clip-style. And clip is the kinase that Stefan actually works on. So I'm not going to really describe all of this thing, but I think this is really to show you that we have absolutely convincingly proved it. And also we have really proved it in vitro. And this is very interesting result in a way that how surface charge complementarity allows the different kinases to really recognize the same substrate but with a different phosphorylation state. And this is the result. So this is the structure that Stefan has really done. Clip. If you really look at it, this is a red patch in here that the substrate binds, and here you have the alternate red and blue. If you look at the phosphorylation of ASF by SRPK1 and CLIC1, you can really see it that SRPK1 can really, we call it hypophosphorylation. So the half of the serum will be phosphorylated. If you have just CLIC to phosphorylate ASF, you see sort of random phosphorylation. The moment you have already pre-phosphorylated ASF, meaning hypophosphorylated ASF, you add CLIC, then you see further phosphorylation in much more efficient manner. And we also really did the GST pull down. We actually showed that CLIC actually interacts with the hypophosphorylated ASF much more strongly than unphosphorylated ASF. And basically, how do you explain it? That's what it is. So that once it gets hypophosphorylated, that means RH1 undergoes phosphorylated, you have the alternate blue and red, positive and negative, and it's binding in here. And now it becomes a perfect substrate for CLIC in the nucleus. And that's why the RH2 phosphorylation is very important for really breaking through the nuclear speckle to bring it to the site of splicing. Right? This is the last slide. It's a little bit busy slide. What it really means in terms of splicing. This is really what's our ultimate goal. Okay, what's this phosphorylation, hyper, unphosphorylated? What is really meaning to the RNA binding? There's no experiment been done in vitro. It's how SR protein binds to RNA, and what's the role of the phosphorylation. And so this is extremely difficult experiment because SR protein are very difficult protein to deal with. So what we really say is that even though the name call is the RNA, this is the RNA binding domain, it actually binds RNA, which is EAC sequence. So this is actually a sequence we know. This is actually a pmRNA in a kinase that the sequence is present, and it's binding the micromolar. So this is micromolar. It's micromolar affinity, anywhere between 1 to 5 micromolar. If you really look at unphosphorylated ASF, it's binding in a nanomolar affinity. It's roughly around 50 nanomolar affinity. If you have hypophosphorylated ASF, it also binds in a nanomolar affinity. If you have hyperphosphorylated, it binds at a micromolar affinity. So we did both this MSA and filter binding, and in the filter binding, again, it's very clear that basically hyperphosphorylated, fully phosphorylated ASF binds RNA very poorly. But unphosphorylated and hypophosphorylated bind very strongly. But only different though, unphosphorylated ASF binds RNA also non-specifically. But if you have hypophosphorylated, the non-specific binding is really gone. If you really look at the filter binding, the presence of large amount of non-specific RNA that hypophosphorylated and unphosphorylated bind in the same affinity. So even though these numbers really, the two different methods that we use, the numbers vary, but the trend is very similar. So now what it really means is the RNA binding. So the RNA binding domain doesn't really bind RNA very strongly. What it really requires the RS domain. So RS domain, we did more experiments, but RS, that to really prove the point that the RS domain actually does something, either directly or indirectly, enhance this affinity of RNA by RNA binding domain. But what it really requires, even the hypophosphorylated can really do it, because so the part that undergoes phosphorylation that doesn't bind to RNA anymore. But you still have the RH2 part that is unphosphorylated, and so that can really allow this enhanced RNA binding. The moment you have the RH2 part is phosphorylated, then you lose this affinity. Now, the last part is really synthesis model. What it really meaning in terms of the spliceosome assembly? What it really means actually that so RH2 actually start with a switch. So if RH2 is unphosphorylated, then it can really enhance its own RNA binding. So it can really, it enhances the RBD's RNA binding in cis. Okay. And the moment it gets phosphorylated, it really opens up. But now it can recruit other RNA domain from other protein, right? Because the spliceosome, or even this SNRNPs, right? U1 SNP, U2 SNP, they have this SR-like protein, and so they can really be attached. So that means that you are really bringing the distal part of the other subcomponent in the spliceosome machinery in there. So by switching phosphorylation and dephosphorylation of the RH2, you can really switch the splicing thing, the spliceosome assembly. And how to prove it? By, if you really can make this just the RH2 part mutated, so the serine cannot be phosphorylated. So you can absolutely, then you can really put it only in the one state, and then really do the splicing. And this is the experiment we are doing. And this is the last slide. So really, this is a very unusual kind. What is the important feature is that electrostatic nature of this substrate and kinase binding and this movement. And I think that's what the driving force, that as the catalysis is happening, then through this positive and negative charge interaction, that really allow the movement of this substrate. And then in the end, it is a very, very strong implication in biology, or even though we don't fully understand it, hopefully somebody will understand it. These students actually started working on it. Brad Nolan worked on the East system. He's now in assistant professor at the University of Oregon. Jackie, who did the ASF and SRP1 crystal structure, who is now an assistant professor at the University of Hong Kong. Sudapa is a postdoc in Germany. She all did a lot of biochemistry on this and the cell biology. Randy worked on the East kinase and the East substrates, so they all left the lab. And really currently the Suyang is a postdoc who's doing all the RNA binding and really has done amazing work. And Nat and Kayla, they're like two master students. They really done very, very nice work. I haven't really described hard work at all. And there I have really very good collaboration. So this is, I started this project because Fu is here. He's a sort of a splicing person at UCHD and Fu's people. And Joe is a geneticist, enzymologist, is really amazing, and he really figured out the mechanistic aspect of the phosphorylation and Joe's students. And Stefan, I just started collaborating with him in terms of click part. And then Andy and Justin, and Andy's postdoc, it's really a very, very nice work. I didn't really describe it because with that Justin's work, I'm completely booked to believing this molecular dynamics, because everything he told me about the substrate and kinase interaction and how it can happen, I mean, they're really all on target. So I thank him for it. And Adrian Kenyon is also the someone I always talk to him to know about the splicing and what he thinks about it, because he's the one actually cloned ASH. And thank you for your attention. Yes.
You have this cartoon of the model. You want to go back to the cartoon of the model? When you say I'm going to show the model of it. Right.
Yeah.
So I'm confused here. So how does it get phosphorylated when it's several inches away from where the site is? I don't understand that.
No. So what I said is that we don't see so the blue part is binding in here, right? And then you have this whole green part that we don't see the density. So we did it. We look at the distance, it's calculated perfectly. It can really
well, the first little red P there is actually coming from the ATP that's in the active site.
No, it's not coming from the ATP. Right? So the phosphorylation, I said that it is in the density, but we don't see the connectivity. And we did a whole lot of experiments with what this phosphorylation is coming from. It is coming from the crystallization condition. We have the AMP, TNT in the crystallization condition. But I think it has ATP contamination. So during crystallization, it was phosphorylating only one serine, the very first one. And that serine gets phosphorylated and binds in this pocket. And we don't see the rest of the green part, the density of it. So we don't see the continuous density. But I think this is what's happening. The substrate, during this, and we have done this mass spec and everything to confirm it.
My question is not so much about the detail of this. Are you saying that when the blue part is bound there,
yeah,
the green part
is
can reach the active
site? Yeah.
It can do that.
It can do that. Yeah, yeah. We have enough ways to do that.
And the green, the yellow stuff in front of it is not obstructing?
No, it's a model. It's not obstructing it. So it's almost in the way if you really look at the cycling. It's just, it's substrate still can bind, phosphorylation still can happen. It's really all this, enough room to those things to happen. And one thing I didn't tell you, it cannot backtrack. So first, it can substrate only can go forward. It cannot come back because once you have the phosphate attached to it, you really create this kind of state that there is a narrow restriction in here in kinase, so it cannot really come back. It has only one way to go. It really just moves forward. And so I think that's another driving force in terms of energy difference.
So you have the structure of what's represented there from the second on the left. That's your crystal structure. Yeah.
That's right.
And do you ever, so what is the evidence that the beta 4 strand eventually takes the RS docking site of the blue strand?
So that's the cross linking. So
okay.
Yeah, that's the cross linking.
Let's keep with the model. Yeah.
Okay.
Yeah.
Okay. So what it is you put a cysteine in here, and then you put a cysteine in the kinase, and only after adding ATP that we really see that beta force, that residue can cross link only in the presence of ATP. But on the other hand, if you have just put a cysteine in here, and this cysteine can really cross into the kinase cysteine without ATP. Yeah.
And do you have a figure for the contribution of the docking domain to catalysis? I mean, does it enhance catalysis tenfold?
So docking domain, the beta 4 docking domain, right?
Yes.
And what happens if you really remove this docking domain or if you mutate the docking domain? In terms of the efficiency, so we have done this experiment. What happens is that if you really do it, it just processivity is completely gone. It still can phosphorylate, and it phosphorylate all the RS domain actually. It can do that, and instead, if you really could make this beta 4 mutant itself, that you don't really see them assembling in speckle. There's the nucleus also. So it has an effect.
Does the rate drop or just the processivity change?
So this is the experiment that Joe has to really to do it, and we haven't really done all of this experiment yet. So I would, this is, and this is just really all these results are from you actually for this RRM dependence of this thing, that I believe that it would have a small enhancement in rate, but I think that it would have really more contribution to the processability. But it may not be like huge difference, maybe let's say 5-fold. But I think that would have tremendous significance in vivo. That's what my guess is. Yeah.
What kind of phosphorylation patterns do you see if you look at the amount of ATP?
You can actually that if so that's the reason that we have 10 micromolar and all those things, sort of knowing that how many serine it can really get phosphorylated, let's say 6 or 7 or 8, something like that. So you're saying the limiting amount. So Joe has done all this experiment, and I'm sure he's going to really talk about it. He actually said a very nice correlation of the amount of ATP to how many serine you can really get phosphorylated. And he has proven a completely different method that the reactionality phosphorylation mechanism in more details, and he will really talk.
I have a question maybe for everybody, but what is the current thinking about how much movement between the N and C loads is required for a catalytic cycle? Because in some structures you see things, regulators or substrate flying across the two domains, which would limit that, well, I believe. And in other cases you don't. And some of them are active and some of them are
in this case, I don't think there's that much movement, because this is a very robust kinase actually. This is amazing that we made so many different mutations in the kinase, it's still really active.
How can you can't tell anything about NNC load movements from that state?
During catalysis that you're saying, that is the,
I think what you just said has no bearing on
No.
the issue of how much NNC loads move.
so what we're saying is that active, inactive, there's a lot of kinases that in inactive, a lot more movement is there. But I'm saying this kinase is already right in the active state, right? So all of these structural features.
But it's moving the kind of two domains moving. I don't know.
But that you don't know for any kinase. You know for any kinase? Oh.
So is anybody doing single molecule things? I mean,
Oh, so like how much open and what happen in different kinases? Okay, so yeah.
Related to that, is this kinase regulated?
The spatial regulation. So that means that it is in the cytoplasmic kinase. It goes to the nucleus in a very specialized time during cell cycle. So the kinase is really the localization is that's what really makes it regulated. So
Because once it gets to work, you switch off the spliceosome machine, don't you? Phosphorylating its substrate stops the spliceosome working. Is that the message right?
no. Spliceosome formation requires both phosphorylation and dephosphorylation. So cycle. So that's more complicated and it's really actually we don't know that. Yeah.
What happens in vivo if you remove the spacer in your kinase range?
It's all in the nucleus. It makes our cells die fast.
So do you see it then as just a changing cellular localization?
So, yeah, so there's my collaborator who has just submitted a paper and did some developments really to addressing that question. So again, it's really connection to HSP, HSP90, HSP70. There's a whole lot of regulation in there. So spacer actually some way involved in regulating the localization of this kind. Yeah.
Your third cartoon, where you have two phosphate nodes,
Yeah.
the second one also came from the active site? The third cartoon, you pointed at the second. Yeah, that phosphate.
Wait, so I think I'm missing something that you're asking, maybe disconnect.
Well, how does it get phosphorylated there? Where's that phosphate coming from?
ATP. It's like any
Yeah, but
kind.
how does it reach the bindings, the active site? Well, the other phosphate is bound in this pocket.
No, the other phosphate is bound to a different part. The phosphate is bound
to the P plus one loop.
to P plus 2 loop. See, this is what it is.
It's right. It's very
Right, so it becomes P plus 2 position, and that
This
is
your cartoon is really not really giving the dimension.
no, not yeah. It's a cartoon.
I'm trying to figure
Yeah.
out.
Okay, yeah.
It seems like it's bound there. How can it get phosphorylated? I don't understand.
When you're being formed, is that the substrate?
Because it's more like substrate.
So
Yeah.
beta 4 is in fact the substrate side.
Oh, I could confuse you all. If kind of people are confused.
Did you try
There is
to
all my.
simulate that with the rate kinetics to see?
No, Angela will do all of those things.
No, but I'm molecular dynamics. I'm just talking about rates to see what's the advantage of having a binding pocket to do something.
What's that advantage? Yeah, we know from the mutational experiment that Joe Lab has done it that actually reduce that this pocket, P plus 2 pocket you're talking about. Yeah, that you reduce the rate of also a little bit of processivity, rate of phosphorylation is reducing. Yes.
But presumably if you bound too strongly to the pocket, then you'd never process. You'd be stuck with the first one.
No, why would we do that? No, that has nothing to do with it. P plus 2 pocket, so let's say first you get phosphorylated. Right after you get phosphorylated, it is that phosphate is binding to the P plus 2 pocket. It has moved. Now the next series is coming in the active site. Now ATP is phosphorylating. Again ATP is transferring it. And now second one gets phosphorylated, displace first one. Okay, now the second one binding to this thing, so it's like that.
To displace it, it has to be not so bound strong.
No, so in terms of the strength of binding, it cannot, eventually it has to get released, right? So I think as every cycle, this is speculation, as every cycle of phosphorylation, I think its affinity getting decreasing, right? More and more and more and more. And so then you really have so much positively charged now, you really have that so much negative charge that it has no affinity. We know that if you fully phosphorylated this or RS1, then it doesn't really have affinity for kinase anymore. Because you started with 50 nanomolar, I think that you're progressively losing affinity.
if you mutate only one of the sites, so this is a sequential process, right?
Yeah.
So if you mutate one of the sites, does it stop?
Joe will tell you all of this thing. He has the, I don't want to
So
say his way. We'll continue.
Where does the RNA bind? My last thing, do you have any idea where the RNA binds somewhere?
You know, I could figure all of this thing out. I have no grant on this thing. I have to tell you the relevance. I started writing from grad, was a graduate student in 1999. 12 times I failed. NIH failed me. And I could have done all these questions you are asking, I had this answer. Unfortunately, I don't have. And because no one believed that I can do anything beyond enough capability. So, so that's just the thing. You know, I think I have given a lot with $0 and the kindness of one person. But it's very unfortunate. But, so.